So let me now discuss a little bit of what's happening in the lateral geniculate nucleus and of the thalamus, as I said. Thalamus is roughly the size of a walnut. It's in a region of the brain that's subcortical, that's called the diencephalon. And there's a lot going on in this walnut-sized thalamus, and not all of it concerns vision. It's a way station for sensory pathways that include somatic sensation and other sensory modalities, and vision is just one. So there is a subnucleus of the thalamus that is specifically related to vision, and it's called the lateral geniculate nucleus, or more properly, the dorsal lateral geniculate nucleus. So here's a diagram taking the lateral geniculate nucleus out. It's just a small part of the walnut, and the organization of this structure is really remarkable. So here you see it in what would be a microscopic slide. The color coding is false, but it is showing you that these layers in this little structure, the lateral geniculate nucleus, are very precisely organized, and they are organized, as shown by the color coding here, into 4 layers that are called parvocellular layers. Parvo means small or scarce, and you can see just from the density and the size of these little green dots compared to the 2 magnocellular layers. Each of these little dots are cells in these layers, that these layers, the magnocellular layers, as the name implies, includes larger cells than the parvocellular layers. And again, you could ask, well, what is that all about? What's it doing? And why is the thalamus, or the dorsolateral geniculate of the thalamus, layered in this very impressive way? Well, one again doesn't know fully the answers to these questions, like so much else in vision, but one can say kind of roughly that these 4 layers, the parvocellular layers, are concerned more with foveal vision. They are concerned more with the precision of high-resolution central vision, and they are particularly concerned with color in a primate like us that has color vision. Not all animals have color vision, of course. We'll come to that later in the course. And these layers are more concerned with grosser change in the image that's on the retina, movement, change in the image. And so, roughly speaking, you can already begin to think of the visual system consisting of different pathways. One pathway, this parvocellular pathway, focused on precision and color vision, and this magnocellular pathway more focused on movement, on change in the image. What's important in this layering, and I think it emphasizes a point that perhaps was obvious to you when we talked about the retina, but the retina is laid out such that the image really exists in the array of photoreceptors that are present there. That is, one photoreceptor is adjacent to another photoreceptor, and those 2 photoreceptors are receiving closely spaced points in the visual space that's translated as an image, as we discussed, onto the retina. So that's a neighbor relationship that you might think is maintained throughout the visual system or not. In fact, it's maintained. That neighbor relationship is referred to as retinotopy. Let me write that down. And it's maintained in the thalamus, and as we'll see, it's also maintained in the primary visual cortex. So if you were to pass a microelectrode, a recording electrode, down through these layers and ask, what's the position in visual space that a cell here is responding to, a cell here is responding to, a cell here or here is responding to, the answer would be that they are all responding to the same point or nearby points in visual space. So the retinotopy that's obvious at the level of the retina is maintained in the thalamus, and as we'll see, as I said, in the cortex, and that's going to be important. So the next question you might ask, well, okay, why is that way station there? Why do you need a way station? Why not just send the retinal information right to the cortex? What's all this thalamic business doing? What's the information processing that's going on there? Again, the answer is not terribly well known. Lots of people, it's not for lack of trying, lots of people have studied neurons in the thalamus and asked the question, what's changed from the information on the retina going through the thalamus and on to visual cortex? And surprisingly, there doesn't seem to be a very radical change. That is, if you record information coming into the thalamus, you can ask into the lateral geniculate nucleus of the thalamus and ask how is it different than the information that's coming out. The answer is that it's really not too different. So the point that does need to be made about the thalamus is that even though the information coming in and going out is not radically different, there's an enormous feedback to the thalamus from the visual cortex. And that information is obviously coming down from the cortex, is obviously doing something. What it's doing, that's where the ongoing mystery lies. But there are about 10 times as many inputs from the cortex back to the lateral geniculate nucleus in the thalamus as there are retinal axons coming up and going into it. So it's processing in a big time way, but no one really quite understands yet what that processing is all about. So coming back to the retina and the magno and parvocellular pathways, you might well ask, is there any difference in the retina that corresponds to the magnocellular and parvocellular layers of the thalamus? And the answer is for sure there is. So these are ganglion cells in the retina. You remember that when we talked about the retina, the ganglion cells are the output cells of the retina that feed into the lateral geniculate nucleus of the thalamus. So it may surprise you to know that there are many different types of ganglion cells. In fact, there are an estimated 60 different types of ganglion cells, if you can believe it, as retinal physiologists and anatomists have worked out. But there are two major types that are related to what we've just been talking about. Small ganglion cells that are called parvoganglion cells or P ganglion cells, and larger ganglion cells that are called magno or M ganglion cells. And as you might imagine from the nomenclature, the large cells are going to the magnocellular layers and the small output cells of the retina are going to the smaller cells in the parvocellular layers of the thalamus. So again, that's by way of saying there is a lot of more precise information transfer going on. Exactly what that means and what it's doing is less well known than you might imagine.